Hello ladies, today I'm going to do a quick review of the cancer unit of cell and molecular biology. Okay, so we have established so far that cancer is a group of diseases and not a single disease, because even though it has certain related characteristics, each and every type of cancer has a different characteristics specific to that kind of cancer. For instance, breast cancer is very different from liver cancer, and so on and so forth. However, all cancers are characterized by uncontrolled cellular growth and division, which mean they lose control of the cell cycle. And we know that cancer is associated with abnormal cellular division. Basically, cells divide when they shouldn't divide, and they lack the normal control systems to shut off this unwanted cell division. So when we see that this is normal mitosis and there is some damage to the DNA, there are a few possible outcomes, one of which is that this damage is so great that it's not repaired and the cell commits suicide or undergoes apoptosis, which is programmed cell death. However, if these mutations continue to persist and the cells accumulate mutations after each and every division, then this can lead to a tumor. So the normal balance between cell death and cell growth is that cell death is approximately equivalent to cell growth. However, in cancer, the growth of cells exceeds the death of cells. Therefore, you have an excess of cancer cells. So you can classify cancer according to various subtypes and subclassifications, some of which are precancerous or leading up to cancer, and some of which are actually cancer. So according to growth type, we have neoplasia, which is the growth of cells to basically form a new structure. So neo means new. Hyper means more. So hyperplasia when there is an excessive number of cells. Dysplasia is when the cells lose their normal arrangement of tissue. Carcinoma in situ is when there is cancer, or basically uncontrolled growth of cells, but it remains in the same place. It's not invasive, it's non-invasive. And finally, we have invasive carcinoma, which is obviously when the cancer cells can invade the surrounding tissue and also can undergo metastasis, which is basically spread through the lymph and the blood vascular system to different parts of the body. So this is normal cells. When there's excessive growth, you see, compared to normal growth, then we call that hyperplasia. In mild dysplasia, we see that the cells start to pile up on top of each other and they have lost their beautiful cell structure or are going towards losing it. Carcinoma in situ is also called severe dysplasia, and you can see there is excessive growth of cells and the cells start piling up on top of each other. They lose their normal structure. However, at this point, the cancer cells have not spread. When the abnormal cells spread to the underlying tissue and to different parts of the body, we call that invasive carcinoma or cancer. Another way that tumors can be classified is according to metastasis, which is the invading capacity. So benign tumors cannot invade and spread to other parts of the body, such as warts and moles, whereas malignant tumors can invade and spread to different parts of the body. So this is basically a benign tumor which cannot spread, and this is a malignant tumor which has began to invade to the underlying tissue and also spread through the blood vascular system. Now, what do cancer cells look like under the microscope? Well, if you compare cancer cells to normal cells, what you see is that there are a large number of very irregularly shaped dividing cells. They have a large nuclear volume, and the nuclei are variably shaped. They're not nice and smooth. They have a small cytoplasmic volume. There's also a lot of variety in size and shape of cancer cells. They also lose the specialized cell features that make them a kind of cell. For instance, a neuron has a specific structure. However, a nerve cell, which has begun to change into a cancer cell, loses that specific structure. Also, cancer cells are highly disorganized, and the tumor boundaries are usually very poorly defined. So if you look under a microscope at a histology slide of a tumor, you'll see that in most cases the tumor boundary is not very well defined. So here we see a normal cell, and you see a cancer cell. You can see this plasma membrane is irregularly shaped. In this case, we have more than one nucleus. We have multiple nucleoli, and you can see that the vacuoles are filled with lipid, so on and so forth. There are several characteristic features of cancer cells. One is that they lose contact inhibition. So when normal cells divide and grow, they stop dividing when a cell will come into contact with another cell. However, when cancer cells divide, they can pile up and divide and keep dividing any which way but what, basically. So they don't have contact inhibition. Cancer cells can keep on dividing even when they come into contact with another cell. Cancer cells also show unrestrained control of growth because they lack the normal control systems, which basically shut off cellular growth or shut down the cell cycle. So you can see here, these are HeLa cells. Also, cancer cells have the property to metastasize or to spread from one part of the body to another. Some biochemical properties of cancer cells is that they have increased glycolysis. So cancer cells prefer to produce energy through the glycolytic pathway just because it is much more quicker and does not require any oxygen. However, cancer cells can switch from oxidative phosphorylation to glycolysis and vice versa, depending on the particular environment of that cancer cell. Cancer cells have a loss or alteration in cytoskeletal protein. So this is a normal cell, and you can see here that the cell scaffolding, or the proteins that basically give the cell its structure and rigidity, are lost or very much reduced in cancer cells. And this is a property which helps cancer cells be more motile or move throughout the body. That's why they are very flexible. Cancer cells also lose anchorage dependence. Normal cells usually have to adhere to a substrate in order for them to grow. However, cancer cells can grow in a suspended culture, and of course this is also an adaptation in the body because cancer cells need to spread throughout the body, and that's why they lose their anchorage dependence. And cancer cells are obviously nearly immortal. So when you grow cancer cells in a cell culture, they will pretty much continue to grow indefinitely as long as you keep on switching out the media. However, normal cells will reach their limit after around 30 or 40 divisions, and this limit is called Hayflick's limit. Now, cancer is basically a genetic disease. So this is the karyotype of a normal cell, and this is a karyotype of a tumor cell. And what you see here is that the tumor cell has lost chromosomes or has excessive numbers of certain chromosomes, and there are also lots of chromosomal translocations. And cancer cells show somatic mutations, okay, somatic mutations in cancer cells. Now that leads us to the question, what causes cancer? Well, there are various different causes. Viruses and bacteria can cause cancer, chemicals, radiation, heredity, diet, hormones, all these are known to play a role or have an effect in causing cancer. So there are a bunch of different chemicals in tobacco smoke that can potentially cause cancer. Low strength radiation can cause cancer, as can high strength radiation from X-rays. Viruses such as the HPV virus, human papillomavirus, can cause certain types of cancer. In this case, HPV causes cervical cancer. Heredity. If you inherit certain mutated genes, you have a predisposition to potentially developing cancer. It doesn't mean that you will absolutely get cancer. So basically what we found is that cancer is a group of related diseases which are characterized by uncontrolled cellular division. Basically, cells divide when they shouldn't divide, and they lack the normal cell cycle control systems to shut off unwanted cell division. Okay, let's now talk about oncogenes and tumor suppressor genes. So, what are oncogenes and what is the difference between an oncogene and a proto-oncogene? Well, oncogenes or tumor genes are genes which have the potential to cause cancer. So the normal cellular genes are called proto-oncogenes. When these are switched on, they can turn into oncogenes which have the potential to cause cancer. Okay, and proto-oncogenes are usually involved in normal cellular growth pathways. However, when these oncogenes are mutated, they can cause cancer. So basically, when a proto-oncogene becomes activated, it turns into an oncogene and it may cause cancer. So how exactly are oncogenes activated? Well, they can be activated by mutation, amplification, or translocation. Mutation. So oncogenes can be activated when there is a mutation in the DNA of that gene. This could be in the form of a point mutation, or otherwise what could happen is that a insertional mutagenesis may occur where a certain gene is integrated with a certain oncogene, and what happens is that that gene will then switch on that particular oncogene. So here we have the ALV provirus, which becomes integrated through insertional mutation with the c-Myc gene. This subsequently turns on the c-Myc gene, and there is uncontrolled proliferation. Another way that oncogenes can be activated is through amplification of oncogenes. So when there's more than one copy of a gene, that is called amplification. So amplification is also another way in which these oncogenes can be activated. And finally, oncogenes can be activated by translocation when there is an exchange of chromatin between two different chromosomes. So an example of this would be Burkitt's lymphoma. So T means translocation, 8, 14. So there's a translocation between chromosome 8 and 14 where the c-Myc gene is placed under the control of an enhancer sequence of an immunoglobulin gene. So basically what happens is that the c-Myc gene, where's Myc? The c-Myc gene gets activated by the enhancer. So we have the enhancer and the c-MYC gene, and the c-MYC gene becomes translocated near the enhancer, which then will switch on the c-MYC gene. Now, what are tumor suppressor genes? They're exactly as the name suggests. So tumor suppressor genes can suppress tumors, and they're basically normal genes. However, their absence can lead to cancer. So basically, when a tumor suppressor gene is mutated, that could potentially lead to cancer. Okay. For instance, p53 is a tumor suppressor gene which produces a tumor suppressor protein, and this particular protein can trigger cell suicide or apoptosis. So in a normal cell, if there's excessive DNA damage, then obviously that could lead to cancer. However, p53 protects the body by causing the cell to commit cell suicide or go into apoptosis. However, if the p53 tumor suppressor gene is inactivated through mutation, then that could potentially lead to cancer. Now, let's talk about the multi-step nature of cancer. So, cancer is a multi-step process. So Knudsen's two-hit hypothesis states that, okay, where is it? A cell can initiate a tumor only when it contains a minimum of 2 mutant alleles. So basically what that means is you need a minimum of 2 hits to cause cancer. Okay, so since cancer is a multi-step process, you can see from this picture that multi-step genetic damage can lead to cancer. So you have this first mutation here, then the cells divide, acquires a second mutation, the cell divides again, acquires a third mutation, acquires a fourth mutation, so on and so forth. And you can see that these genetic mutations in the genotype are basically correlated to phenotypic characteristics. You can see as the mutations accumulate, the cells become more and more deranged, right? And normal cells can evolve into tumor cells through a progression called tumor progression. So we're very familiar with this figure by now. So you can see the progression of a normal cell turning into an invasive cancer. And this is accompanied by mutations in various genes. For instance, there's a loss of, this is an example from colon cancer. So in the normal epithelium, there's a loss of the APC gene, followed by the activation of an oncogene called KRAS, then the loss of a portion of a tumor suppressor gene, then the loss of p53, which is also a tumor suppressor gene, which can ultimately lead to cancer. So basically, the phenotypic features, or basically the genotypic mutations, are mirrored to the phenotypic alterations. So as we know, cancer is a genetic disease, and all these somatic mutations lead up to cancer. However, cancer is a multi-step process and therefore requires a number of steps before that deranged or abnormal cell actually becomes cancerous. Now let's talk a little bit about the cell cycle. So what is the cell cycle? Well, it's basically the cell division cycle of mitosis. So it's basically a programmed series of events that enables a cell to duplicate its contents and generate 2 daughter cells. Basically, the cell divides to give 2 daughter cells. This is the mitosis phase, and G1S and G2 is the interphase. Okay, so G1 is the post-mitotic gap phase, S phase is the DNA synthesis phase, and G2 is the second gap phase. Now, what would happen if a cell which has its DNA damaged goes through the cell cycle? Well, that's why we have checkpoints. There are checkpoints at a number of places in the cell cycle that check whether the cell is damaged or not. And accordingly, it will either get it repaired or cause the cell to commit suicide or apoptosis. However, if this damage is neither repaired nor the cell dies, then this damaged DNA persists and could lead to cancer. Okay, so let's talk about the where did it go? About these various checkpoints. The main checkpoint is in G1, and this is called the G1 phase checkpoint, or the restriction point. So if a cell passes through the restriction point, then it is committed to divide. So if there's some DNA damage in a cell at this point in G1, then it still can be repaired. However, if this passes through the restriction point, then the cell is committed to divide. Okay, so let's skip over this part. So what do we know about the cell cycle? Well, the cell cycle is a programmed series of events that enables the cell to generate two daughter cells. There are various checkpoint controls in the cell cycle. The G1 phase checkpoint is the most important, and it is called the restriction point. And once a cell passes through the restriction point, it is committed to divide. And how do cancer cells override these checkpoints? Well, they try to inactivate one or more of these checkpoint controls in order to cause cancer.